Get started. So it's my pleasure to introduce today's speaker, David Bedwell. He's a professor and chair of the Department of Biochemistry and Molecular Genetics at the University of Alabama, Birmingham. He's also the co-director of Structural Biology Program and the associate director of the CF Research Center at UAB. His laboratory is focused on developing therapeutic strategies for genetic diseases caused by mutations that introduce premature translation termination codons. His group is working to identify drugs that suppress premature stop mutations in the CFTR gene as a treatment for CF. These treatments allow translational read-through to generate full-length functional proteins, and this approach may be applicable to polycystic kidney disease. So we welcome David to the group, and his title of his talk is Distinct Classes of Small Molecules that Suppress Non-Sequence Mutations that Cause Genetic Diseases. So thanks, David.
Okay, thank you for the introduction. So let me just go ahead and hit my disclosures real fast. So I have funding from the CF Foundation, the Gilbert Family Foundation, and the NIH. All right, so let's jump right in. So I'm going to start with one slide of the big picture, a bunch of different ways that you could, hypothetically treat nonsense mutations. And then I'll zero in on the way we do it, or at least the way we're trying to do it, which is small molecules. So the different major classes are gene editing. This is an approach where the primary mutation is corrected in the genome so that the proper protein expression and localization and function is restored. So this is the kind of the gold standard. If we can do this routinely, we could cure a lot of genetic diseases. Another alternative approach for gene editing is to edit an endogenous tRNA gene to make a suppressor tRNA. David Liu at the Broad Institute recently published a Nature paper with this approach, and so this is viable. If we move on to gene and RNA therapy, so gene therapy is with DNA, RNA therapy is with RNA. So in either case, we introduce a wild-type copy of the mutated gene or its mRNA, and we can also introduce a suppressor tRNA gene or a suppressor tRNA directly. Now, both of these, the editing and the gene therapy, have various challenges. So for gene delivery, you need to target directly specific cell types, in particular stem cells, so that it can be transmitted, it's basically you're correcting the stem cells so that all the subsequent cells that derive from those stem cells are corrected. With mRNA therapy, you're going to have to do repeated delivery, and that's complicated. But the good news is you don't have to hit the stem cells then. You can hit the cells that are specifically missing the functional protein. All right. And then with all of these things, either the targeting with the editing or with the gene therapy, it's possible that you could actually mess other things up. In other words, disrupt another gene or something like that causes issues. And as most or all of you know, there are examples of people getting cancer from various gene therapy approaches in the early days. All right, so we've taken an alternate approach. And as I said, if we could edit the gene and fix it, that's the gold standard. That's where we all want to go. But we've been looking for either gene therapy or gene editing for decades now, and it's not here yet. And they're making progress, but I think it could still be quite a while. So what we're looking for is an interim approach with small molecules so that we can pharmacologically suppress nonsense mutations, or as I define here, premature termination codons are in the mRNA are called PTCs. So these can alter the function of either ribosomes or the translation termination factors to reduce termination at PTCs. This read-through process, as it's called, restores synthesis of the full-length protein, even though the PTC is still present in the mRNA. And then, as I mentioned, for the gene editing and gene therapy, there are caveats, there are issues, and I will address those throughout the rest of the talk. All right. All right. So initially, let's talk about translation termination and how it works. So here we have a ribosome in a pre-termination complex. So you have the peptidyl tRNA in the P site. You have a stop codon in the A site. And there will be random sampling of tRNAs and termination factors into this site because the cell doesn't know what the codon is at the base of the A site. But when a termination complex comes in, which includes ERF1, ERF3, and GTP, that can bind to the A site. GTP hydrolysis by ERF3, it's a GTPase, leads to a conformational change of ERF1 that allows accommodation of ERF1 in the ribosomal A site, and that leads to polypeptide chain release. And then we end up with a post-termination complex that gets subsequently disassembled and recycled for another round of initiation. All right. So this is basically telling you everything you need. The only other thing that I'm going to talk about today that's not on this slide is at the base of the A site is the decoding site, which monitors either codon anticodon interactions or stop codon ERF1 interactions, okay? And that's the site where aminoglycosides bind. So aminoglycoside binding affects fidelity of translation, and then the abundance of the termination factors can also affect the amount of read-through that occurs. All right, so people years ago did meta-analyses of all the known genetic mutations at that time, and they found that about 11% of all mutations that cause inherited human diseases are in-frame nonsense mutations, okay? So based on this, it's reasonable to assume that approaches to suppress nonsense mutations could provide a therapeutic benefit for patients with many genetic diseases, PKD, CF. I'll talk about neurofibromatosis, but if you think about it, any disease caused by a loss of protein function, there's going to be a genotypic subset of patients with a nonsense mutation. All right. Okay, so how does this work? So upper left, we have a nucleus with a gene that's got a premature stop in it. This is the primary transcript is made by Paul II. That is cap spliced, processed, polyadenylated, and then that mature mRNA comes out into the cytoplasm to undergo what's called the pioneer round of translation. And two things can happen to this mRNA. The first is if you translate this pioneer round, you will come to the premature stop codon and prematurely terminate translation. And that leads to a truncated protein that's invariably non-functional or unstable. And that, in turn, this premature termination frequently will lead to a process called nonsense-mediated mRNA decay, which leads to a significant reduction in the abundance of the corresponding mRNA that's encoding this premature stop codon. Okay, this decrease can be as little as twofold, it can be as much as 20 to 30-fold, okay? But these are things that complicate the process of anyone trying to suppress nonsense mutations. Okay, so the key here is if you zero in on the termination process, the premature termination process, the great majority of time, the ternary complex with ERF1, ERF3, and GTP will bind to the stop codon and facilitate termination. And let's just call this 99.9% of the time. However, this termination process is in equilibrium with near cognate tRNA misreading. And this actually happens all the time. And with our sensitive luciferase-based reporters, we can actually detect low-level read-through of reporters all the time. okay? And let's just call it 0.1%. So what we're trying to do with this approach with these small molecules is skew this equilibrium so that termination wins a little bit less and the near cognate read-through occurs a little bit more, maybe 1%, maybe 10%. And that hopefully is going to give you enough full-length protein with function to cause a therapeutic benefit for the patient. And read-through compounds that most people have heard about include aminoglycosides like G418 and a protein that's, a small molecule that's been around a long time called atalurin, or its old name was PTC 124. Both of these compounds have problems. Aminoglycosides are toxic. Atalurin is not very effective. And I think I've read recently that PTC Therapeutics is no longer pushing the atalurin for clinical trials. All right, so this is what I'm going to talk about today. So here we have a ribosome with a peptidyl tRNA in the ribosomal P site, a stop codon on the A site. Here's the translation termination complex. And we're going to talk about a handful of different mechanistic classes of read-through compound. The first are the old granddaddy of all read-through compounds, which are aminoglycosides. G418 is the one that's most commonly known. ELX02 is a synthetic aminoglycoside that was selected to be less toxic than other natural aminoglycoside antibiotics. There's also a class of compounds that us and other labs have identified that are called aminoglycoside enhancers. Sorry, this thing keeps jumping on me. All right, and then we have two classes of molecular glues, it turns out. One is ERF1 degraders, and these facilitate degradation of ERF1 by gluing ERF1 to the ribosomal A site, inducing ribosome collisions and inducing the ribosome quality control machinery, and that leads to degradation of ERF1. The other class of molecular glue includes ERF3 degraders. These small molecules will bind ERF3 to an E3 ligase receptor called cereblon to facilitate its ubiquitination and degradation by the proteasome. So both of these are mediated by the proteasome. Okay, so that's basically, I'm going to talk about ERF1 degraders, ERF3 degraders, and aminoglycosides today. All right, so first ERF1 degraders. So these were identified initially about almost a decade ago. A high throughput screen with over 750,000 compounds was carried out. We found two classes of molecular glues that cause the programmed degradation of ERF1 through the RQC pathway. These compounds aren't particularly potent themselves, but they are substantially augmented by the presence of low-dose aminoglycosides like G418 or ELX02. One of these compounds, SRI 41765, exhibits activity in primary HBEs. These are human bronchial epithelial cells, and everything is directed, or most things will be directed towards CF because CF is paying for these studies, the CF Foundation. And when we add 41765 with a low dose, such as an EC10 of the aminoglycoside ELX02, we can approach clinically relevant CFTR activity being restored in these bronchial epithelial cells. And so 41765 is one of the classes that we have that we have developed further to pick up drug-like properties, including solubility, permeability, animal PK, and a lack of toxicity. All right, so here's an example of what we're doing. So we start with a primary hit from the HTS. We take the scaffold and we start walking around it with our medicinal chemist, Corinne Jelly Zafran. We'll walk around the structure and modify it. And what we ultimately get is more and more activity. And you can see it here is over on the left. The red is the G418 EC10 alone. And as we move across, we can get a significant amplification of that activity. Let me see if I can get rid of these. There you go. All right. So these are two of the better ERF1 degraders that we have. And we'll talk about primarily 41765, but also 41819. Okay, here's a little functional data with a nanoluciferase assay. Here's the activity on its own. And then we add the G418EC10. We can go from, 10-fold increase to 200-fold increase. So we get a synergistic response in the read-through activity. This, in the middle part, we're just showing transepithelial chloride conductance assay. So this CFTR is a cyclic AMP activated chloride channel. So we can stimulate CFTR activity in a monolayer with addition of forskolin here. And then we measure the activity, and then we hit it with a CFTR inhibitor here, and we determine the area under the curve. And we can see that there is modest activity with CFTR alone, with 41765 alone, and it's much higher when we have it just a whiff of G418 here. Okay, and this is done by Steve Rowe's lab. So our group, the three labs that are working on this project are my lab, Corinne's lab, and Steve Rowe's lab. All right, the ERF3 degraders. So we have been pushing very hard over the last year or two on these. We are again testing them in our luciferase assay, as well as the CFTR tech assay. As I mentioned before, these are molecular glues that bind ERF3 to cereblon for ubiquitination and degradation. And we have a number of these that are showing pretty strong activity, greater than 600 or six-fold the G418 EC50 activity we see in combination with a low level of G418. And we are currently working on optimizing these derivatives to, again, make greater solubility, permeability, and all the things you need to make a drug. So here's an example. These are the left half of this was where we were about 4 to 6 months ago. And since then, we've continued to get better and better compounds. And so we now have something like 9 compounds that are greater than 6-fold the level of G418 EC50. And G418 is always our positive control. So now the question is, what is the amount of compound required to get these activities? And so if we take these 9 top compounds and plot them as a function of their EC50, we see the 9 compounds here with the red arrows, and here's our 2 older ones that were the best earlier on. They're about half millimolar potency, and these you can see are down in the 10 nanomolar range. So we have now compounds that are quite active as well as quite potent. And these are the ones we're going to process further to develop drug-like properties and move forward into clinical trials. All right, just to show a little bit of data, here you can see ERF3. Here you can see ERF3. When we throw in a relatively high dose of this compound, ERF3 disappears. So, of course, we're giving these high doses because we're trying to look at the maximum activity we can obtain. You see a little bit of decrease in ERF1. This is the complex between ERF1 and ERF3, and ERF1 is not as stable when ERF3 is missing. Darn it. Over here in B is CFTR activity, and you can see we're pushing up to 15 or 20% of wild-type CFTR activity. This is a quantitation of the Western blot. And then here's a quantitation of the ERF3 protein abundance. Okay, so we are seeing an increase in CFTR abundance and a decrease with ERF3 abundance. All right, so as you can see, in many cases, we are challenged because CF is a difficult disease to work with because 30 to 35% of wild type activity is required to provide a relevant therapeutic benefit for CF patients. And what I'm referring to is lung function assays. All right, so we made the premise that individual read-through compounds may not induce enough read-through to restore the level of CFTR function needed to provide this high therapeutic benefit. So we made the hypothesis that combining different mechanistic classes may provide additive or synergistic increases in read-through. And this is not unlike the approach the pharmaceutical company Vertex made when they actually developed a triple combo modulator therapy for people that have trafficking defects, folding and trafficking defects in the CFTR protein. So Trikafta is best known. It's got two compounds that are called correctors that help folding of CFTR in the ER so it can get to the cell surface. And the third compound is a potentiator, which actually helps the channel to gate more frequently and provides greater CFTR chloride transport or chloride movement. It's not a transporter. Okay, so this was the precedent. It's accepted in the CF field, and these compounds, this Trikafta triple combo and the Aleptric triple compound, which is a newer version, these are significantly extending lifespan of CF patients, and it's predicted that they will be living decades longer now with these compounds. These are not effective for patients with nonsense mutations because they bind to the protein to help folding and channel opening. So we have to get protein made before we can even try these. Okay. So, but the concept of combination. So here is an EC50 of an ERF1 degrader. We get about 50% of G408 EC50. Here is a ERF3 degrader. We get about 80% of the EC50 for G408. And when we mix these same two EC50s together, we get a 400% recovery of activity. So this is significantly more than the additive sum of these two. So we are calling this synergy. Okay. So why is this happening? We've done Western blots on this. Here's ERF3. You can see with no decrease with the ERF1 degrader, decrease with the ERF3 degrader, and more decrease with the combo, as quantitated here. Here's ERF1, and it continues to go down and goes down further with the combo. And so what is apparent here is that by combining these, we're getting a greater decrease in both ERF1 and ERF3 than either one by themselves. So we think we're on the really steep portion of the curve, and by pushing the concentration of these release factors down, it's causing a significant greater increase in read-through. Okay, now what about the aminoglycosides? As I said before, they're the oldest known class of read-through compounds. People have, I think Joshua Lederberg and Saul Spiegelman published separate papers in 1964 showing streptomycin could suppress nonsense mutations in E. coli. Some aminoglycosides like G408 show strong efficacy in diverse mammalian cell types, and but the problem is they're only active at high, which are approaching millimolar doses. And these high doses can cause severe side effects, including ototoxicity or hearing loss and kidney failure. And this makes sense because there are cationic transporters that bring aminoglycosides into these cells in the hair cells of the inner ear, which results in ototoxicity, and in the proximal tubules of the kidney. And it turns out megalin is highly enriched in these two cell types, and that's what leads to this toxicity. All right, so what we asked, though, is could we identify aminoglycoside enhancers that enhance not only the activity, but could it also work at much lower doses? So we did another high throughput screen of 500,000 compounds with an EC20 of G408 present, and we identified compounds that increase the activity. And so here, I think it's important to emphasize that if this were the aminoglycoside and we were needing close to a millimolar to get good read through, we're trying to push it down over this way. So that a much lower dose could give approaching, the same levels or maybe even higher. And that's exactly what we saw. So here is the aminoglycoside ELX02. You see at 1 mg per mil, 1 millimolar, we're starting to bend up, which is a very high dose. When we add our aminoglycoside enhancers, either one of these two, they don't do anything by themselves, but they push the curve over so that we actually get more read through at a 50 to 100-fold lower dose. Okay. And so now this activity is shown with an EC10 of ELX02. And this is hopefully low enough that we can do something without causing nearly the level of toxicity as before. All right, now let me switch gears for the last few slides here. We are also applying some of these compounds to another disease called neurofibromatosis, which is funded by the Gilbert Family Foundation. And so neurofibromatosis is caused by defects in NF1, neurofibromin. This is a RAS gap in the RAS signaling pathway. So what RAS gaps do is they bind to RAS. When RAS binds GTP, it's activated. And what the RAS gap does is it binds and stimulates hydrolysis of the GTP to downregulate RAS activity. So when you're missing NF1, as in these tumors in NF patients, the RAS is hyperactivated and you get these neurofibromas, these tumors that are characterized by unregulated growth because of activated RAS signaling. Okay. And so there are mouse models that are good tumor models available, but it takes months to develop the tumors. And then, it's just a very slow process. And so what my collaborator, Bob Kesterson, did is he made a conditional knockout of NF1 so that we can make a faster turnaround in vivo of activity. And so the way this works is he put in, there's 2 copies of NF1. One is a wild type copy that's floxed, and the other allele is a PTC allele, R816X. So when we inject the tamoxifen, we excise the wild type allele, and within 12 to 14 days, the mice die. So this gives us a fast assay so we can determine which compounds are working the best, optimize dosing, and then once we have those in vivo parameters defined, we can go into the longer tumor models, the slower tumor models. All right. So here's an example of a mouse that's treated with tamoxifen and just vehicle alone for the aminoglycoside ELX02 PBS. And you can see the half-life is about 12 days. When we add ELX, we get an extension to about 15 days. So it's a 25% life extension. Here is another vehicle for the 41765, the ERF1 degrader, which is similar, about 13 and a half days instead of 12 days. When we try the 41765, we extended it to 17 days, which is a 26% life extension. And then when we combine the 2 compounds, we went out to 19 days, so there's a 41% life extension. So this is kind of a layout of how we plan to now start testing systematically these things. This is the first experiment we've done with these, so this is preliminary data, but it's kind of tells you the way we're going to try to optimize dosing, the compounds themselves, and any combinations that we might try. All right. So before I end, let me talk about a couple of questions I get every seminar that I give. So I'll hopefully head off these questions or stimulate further discussion. So the first question is, what do these things do to normal termination codons? Are you reading through all the termination codons in the cell? And the second question is, what are you going to do about nonsense-mediated mRNA decay? In other words, can you boost up the level of mRNA back to normal? All right, so first is normal termination codons. So in collaboration with Rachel Green at Johns Hopkins, we did ribosome profiling, and this is not unlike RNA-seq. If you don't know what it is, where RNA-seq, you fragment the RNA, the total RNA, or the poly-A-selected RNA in usual cases, and then you sequence it, and you can quantitate the abundance of mRNAs in the cell. In ribosome profiling, you isolate polysomes, shear and nuclease RNase treat so that only the ribosome protected fragments remain. Then you purify those fragments and you sequence, and you can actually see where the ribosomes were on the mRNA. And you see this in a periodicity of 3 bases because there's 3 nucleotides to a codon. And what we found was, here you see this herringbone, this sawtooth pattern of every 3 bases is a codon. And then here is the termination codon. There's a long pause at termination codons. With DMSO, our vehicle control is here. G418 causes a lower peak. That's the aminoglycoside. And an ERF1 degrader causes a longer pause. But when we take this portion here and expand it, what we see is DMSO and our ERF1 degrader are superimposed, so there's little or no read-through of the normal termination codon, whereas G418 does cause a low-level read-through. So this looks big, but if you look on this, it's actually still very low. So the bottom line is, this has been shown with suppressor tRNAs as well. So normal termination codons are resistant to read-through. And that's probably because they've had, billions of years to evolve to have multiple layers of protection against read-through. And in particular, there's a spatial context that's important. There are factors that are on the 3' UTR, and in particular, the poly-A tail, poly-A binding protein actually interacts with the translation termination factor ERF3 to stimulate polypeptide chain release. And if you have a premature termination codon far upstream, that interaction won't occur, but it helps protect normal termination codons. Okay, so we don't think read-through of normal termination codons, at least in this meta-analysis of 5,000 or 6,000 genes, is going to be a problem. We can't exclude the possibility that there might be a handful that are read through, and then we'll have to contend with that if it causes a problem. The other thing is NMD. So we made reporter constructs that have nanolook fused to beta globin with introns to induce NMD. And then we basically started using various read-through compounds, and they were all clustered down here. And we couldn't really tell much of what was going on. But then as we combined more classes, a combination of 2 classes, 3 classes, et cetera, we get more and more read-through. And what we saw was that the nanolook mRNA actually increased several fold. So this is consistent with a wild-type mRNA not being subject to NMD because the ribosomes are transiting down the mRNA, and they're probably plowing off NMD factors. And the more ribosomes we can induce by read-through to plow through the previously untranslated region, of the open reading frame, should also reduce the level of of NMD as we see here. Okay. All right. So the overall summary, we've identified multiple classes of mechanistically distinct read-through compounds that seem to be working in various CF and NF1 relevant models. Our ERF1 degrader 41765 shows activity in both human bronchial epithelial cells as well as organoids when combined with the aminoglycoside ELX02. The ERF3 degraders are much more potent and more active, and we are seeing significant read-through with those as well. We can get much higher levels of read-through with these than we can with the ERF1 degraders. And then when we combine those 2 classes, it goes up even more. And then I showed you a little bit of data with the aminoglycoside enhancers. We also have ERF3 degrader enhancers, which I didn't talk about, that are also in development. So The bottom line here is that we are trying to develop different approaches with these small molecules to maximize the effect of the read-through agent to restore the maximum amount of the missing protein and its function. Here are the people that did the work. My lab, Kim Keeling's lab, works very closely with me. Corinne Aljeli Zafran is the medicinal chemist who does all the chemistry along with Omar. Steve Rowe's lab. Steve started out with us. He's a professor in the Division of Pulmonary Allergy and Critical Care Medicine. He was actually recruited to be the chief scientific officer of the CF Foundation a few years ago. And then my NF1 collaborators, Deanne Wallace and Bob Kesterson, also participated in our NF1 project. And with that, I will take questions.
All right, thank you, David. There's some questions in the chat. Maybe we can start off with those. Bindu Panda was asking if the ERF1 and ERF3 degraders act both in free form and in complex form.
You mean, will they degrade the complex of ERF1 and ERF3 as opposed to individually? Is that what the question is?
Well, I think it was emphasized on the free form or in complex.
You're right. Okay. So we don't know. These things will form the complex. They're largely in the complex in steady state in the cell, but we can't say for sure that they only work in the complex or separately, or it could be both. We don't know.
Meng Chen was asking, could you demonstrate restoration of full-length CFTR protein by ERF1/3 degradation compounds? Do these read-through compounds also affect the natural stop codon? I think you actually may have.
Okay, so yeah, we've got a lot of data, so we can see. So CFTR band B is in the ER form, band C is a highly glycosylated form that gets to the cell surface, and we are restoring band C CFTR. And so as far as the natural stop codon, I talked about, the ribosome profiling to show that they don't seem to be affecting most normal stop codons.
Okay, just wondering if these compounds might act differentially for different genes with premature translation codon or show uniformity among different genes.
So we think the major effect so the context is important, but it's primarily the local sequence context, a few codons on either side. And these are, within the regions that the ribosome is sitting on. We published a paper in Nature Communications a year or so ago where we actually, a student in Alan Jacobson's lab, did bioinformatics she came up with an algorithm to predict which contexts lead to read-through, what parameters. And it looked like primarily we could predict susceptibility to read-through with as little as a couple of codons on either side of the termination codon, okay? And that was with a 75% probability that we could predict that with just a small local sequence context. So it's really kind of, surprising, but it makes sense because that's where the ribosome is sitting, and it's got to be interactions downstream in probably the mRNA tunnel, as well as upstream in the P site and the E site of the ribosome.
Okay, one more from the chat, and then we'll take some their hands up. Matis Caprizzi said, I take that CFTR assay you utilize are oozing chambers. Did you observe any unusual epithelial leakage upon application of your compounds?
Okay, so you could use oozing chambers. In these studies, we use transepithelial chloride conductance, or a tech assay. This is very similar to an oozing chamber, but it's much, it's higher throughput. So you can do 24 samples at a time as opposed to a maximum of like 8 with an oozing chamber. It gives you, similar information, a similar current trace, and you can quantitate in a similar way. Leakage, of course, if we, add enough compound, we can kill the cells and they get super leaky. So, we have to be careful about, titrating it to a reasonable level. And that's why we were showing, what we routinely do is show the resistance, because when the cells get leaky, the resistance gets very low. Okay. And so we make sure that the resistance is within a certain range before we, otherwise we have to back off on the compound.
Okay, Bino, do you want to ask a question?
Sure, yeah, I thought that was really interesting. And, we're all thinking about polycystic kidney disease, and there's many different nonsense mutations that can cause PKD, right? So it's a wide spectrum. So I think that question about, whether there is a bias of certain codons versus others is an interesting one. So one thought was like, the ELXO2, for example, the aminoglycosides tend to favor the GA versus the AA at the end of the stop codon. Do you see effects of that, of your degraders on that bias at all?
Well, so the deal is termination is mediated by a tetranucleotide, actually. So it's the stop codon plus the first base after. And what happens is that first base is actually pulled up into the A site where it actually, the base is base pairs with a ribosomal RNA residue. And so that actually helps. And purines are more efficiently recognized and terminated in that fourth position as opposed to pyrimidines. And so the world's leakiest tetranucleotide is UGA followed by C. Okay. Whereas UAA is usually much more efficient. And, if it's followed by a purine, that's the best.
Yeah.
Okay. So it's kind of weird, but people have solved the crystal structure of a terminating ribosome, and it's actually a tetranucleotide that's recognized by the release factor. So you learned something new today, if nothing else.
The other thing I was thinking about was, this last set of data looking at the normal termination codons. It wasn't clear to me whether you were looking at the combination there of, ELXO2 or G418 with the degrader compounds, the RF degraders, because that would seem to be the one that is the most clinically relevant here, to be checking for whether there's read-through at the natural termination codon.
So we did that experiment when we were first publishing, our ERF1 degraders, and obviously we just wanted to see what those guys were doing. We probably need to go back and do a more systematic study now. That was an old ERF1 degrader. We need to do it with an ERF3 degrader, which hasn't been done, and just kind of make sure we check all the boxes there, but we haven't done that yet. There's a lot of stuff mechanistically that we would like to understand, so we spent a few years doing tandem mass spec, figuring out what amino acids go in for each stop codon and in each context. And it turns out with the same stop codon, the flanking regions, just a few bases on either side, can alter which amino acid and which amino acyl tRNA is used to suppress. It's incredible.
Very interesting. Thank you.
Brad, I know one guy here.
Thank you for a great talk, David, as always. So, if you look at CF, you look at PKD, it's multi-tissue impacts, right?
Right.
Do you know whether these compounds equally function in different cell types?
Well, it seems like a better way to approach that would be to look at the distribution in tissues, right? They do work in multiple cell types, but we are now looking at for example, with our degraders, we now harvest, 6 or 8 organs from a mouse that's been treated, and we look at ERF3 abundance, and we can show ERF3 going through the floor as we titrate in more and more ERF3. Certainly not every organ. For example, ERF3 degraders don't cross the blood-brain barrier. So the brain is totally normal, but other tissues are showing big decreases in the release factors.
Hey, Dave, we have a question in the chat. Menu, really great talk. I noticed that the dose response curves for your aminoglycoside enhancers did not appear to reach maximal effectiveness, top of the curve. So could you please explain why this is the case?
So in
The
other
other
words,
question is,
we didn't get all the way up to the plateau, right?
Right.
So there's 2 reasons for that. One could be toxicity, or the other could be operator error. He didn't titrate high enough. And either one is equally plausible.
Let's see. Yeah, what effect is this likely to have on the final potency values if you haven't reached the maximum?
Yeah, so we crunch this and let the math tell us what the EC50 is, and that will vary depending upon how good your curve is. So what we really shoot for is getting multiple points on the linear portion of the increase and at least plateauing off, maybe not all the way up, because those frequently will, could go into toxicity issues, which then can screw up your results even more. So it's kind of a trade-off, right?
Okay. Any other questions for David? All right, well, thank you very much, David. I appreciate the talk. It was really interesting, and I think we got a lot out of it. So thank you very
All right.
much.
Thank you.